Thank you, Craig, for that great presentation. We will proceed with the Q&A. The first question is, if I am using expensive material, antibodies or oligos, how do I maximize conjugate yield with the HiNiC and 4FB chemistry?
Yeah, certainly. Number one, as I was just talking about some of the do's and don'ts there, what you want to do is use a highly purified material. Certainly, as I mentioned, for oligos, pass it through HPLC, or indeed you'd want to desalt it as well. And in some of these complete kits, there are things called Zebra columns that get it into a buffer format that make it more appropriate for bioconjugation. So you want to get rid of the contaminants, the preservatives, do a buffer exchange to get it into a good buffer initially to perform that bioconjugation. Make sure also that these particular biomolecules are within the appropriate concentration range. Certainly if you use too little, you might not get appropriate labeling. If you use too much, that's going to be problematic as well. There might be issues with solubility. It may crash out a solution. Another point would be volume as well. Don't use very small amounts, because otherwise you just might lose the material when you go through a purification step at the end, trying to remove the unincorporated, or indeed using too much material. You've just got everything slopping around there, and you might reduce the labeling efficiency there. One other point I didn't really touch on in the presentation was that with the HiNiC and 4FB linkages, there are actually what we refer to as checkpoints during the bioconjugation methodology. And what you do is you check the MSR, the molar substitution ratio, which is really, you check how much HiNiC is attached to, for example, the antibody, and you check how much 4FB is on your, for example, enzyme, that same example I've been using, and make sure you've got the appropriate, molar substitution ratios of those molecules on those given entities before you continue with the bioconjugation. Because that's the great thing about the SoluLINK chemistry, they're midpoint checks to make sure that, yeah, okay, I've got, my estimated, 3 to 5 4FB on my enzyme and 3 to 5 HiNiC on my antibody. All signs point to a successful conjugation, and therefore you move forward, and then you can use that hydrazone bond or chromophore to just again confirm the conjugation has occurred, and you can quantify that amount. So they're the sort of points I'd look at if you're working with expensive material or maybe precious material.
Great, thank you. The next question is, what is the difference between the protein oligo conjugation kit and the antibody oligo kit?
Yeah, I didn't really expand on that in the presentation. So as you saw for both the oligonucleotide kit, we've got an antibody and a protein oligonucleotide kit, and also for the biotinylation kits, we've got an antibody and a protein biotinylation kits. And that does pose the question, okay, if I'm not using an antibody, I can use the other, another protein for the other kit. And the thing is, they are different a little bit in the way they are set up, in that the antibody kits have what I would say more of a highly stringent sort of step in terms of the purification, because we know that antibodies are, around 150 to 160 kD in size, and therefore we include in those kits appropriate cleanup steps to make sure the material at the other end is highly pure. So with protein, oligo kit or the protein biotinylation kit, it's a little difficult to identify what given protein you might be using. It could be much bigger than an antibody, or it could be smaller. And so therefore it's a little more difficult to include in there a given cleanup step for that. So it gives you a little bit of an insight into those the differences, I should say, between those two kit formats.
Okay, great. The next question is, why should I choose SoluLINK over other bioconjugation kits such as LightningLink?
Yeah, fair enough. You know, I didn't touch on other technologies that are out there in terms of bioconjugation. I did show that there are other ways to perform protein conjugation with that abbreviated table. We looked at sulfhydryls and things like that. But I will say that, you know, the LightningLink technology certainly does highlight how quickly it can be done. Interestingly, though, there's no cleanup step at the end. They put in a quenching reagent. But as I pointed out there, I think, you know, if you're working particularly with precious material or you're trying to maximize labeling efficiency, you really need highly reproducible results. I'd highly recommend performing a cleanup step, such as what we recommend in some of the methodologies for the SoluLINK chemistry. But again, what I've stressed throughout the presentation here for the SoluLINK chemistry is the use of the Heineken 4FB, and the fact that they enable one, as I just spoke about in an earlier question, this mid-checkpoint molar substitution ratio check, but also that hydrazone chromophore that allows you to do a quick UV scan to quantify that, yes, the conjugation is successful, and yes, I do have a known amount of whatever it is, biotin, for example, on my given protein or antibody. And that, I think, is invaluable for investigators who want that consistency, that reliability, that reproducibility between manufactured lots that they're doing in their lab. Otherwise, it's a little bit of a crapshoot with some of the LightningLink stuff, and particularly if you know, you're not using highly purified material, there could be a lot of problems with background and inconsistency in labeling, poor labeling efficiency, and things like that. So that's why we incorporate, you know, things such as these zebra columns for cleanup as well. Also, we'll say that we do have some online calculators and other tools and resources that I think would be helpful for investigators to look at as well.
Okay, great. The next question is about siRNA antibody conjugates. Can you elaborate on this application and how do you remove excess siRNA?
Yeah, fair enough. So I did touch on that a little bit, you know, and that was with the, you know, oligonucleotide kit. I spoke about targeted oligonucleotide delivery, whereby you'd use one of those kits. Maybe you'd have a protein, for example, and basically what some investigators are looking at with the siRNA is trying to increase its longevity. Once injected, I think it gets cleared pretty quickly by either kidneys or liver or something like that. And you know, the idea is to apply it, conjugate it to a given target protein. So it can then be incorporated into a cell or cells, the target cells, and then it can do its business in terms of breaking down, you know, complementary RNA. So introducing them has been an issue, and their longevity in this, as in when delivered, you know, systemically. So I think investigators are looking at things called protein cages. I think you can probably look that up in some of the methodologies that are there. And certainly, I think those protein cages act to protect that small interfering RNA from getting gobbled up by the body and allow significant or substantial amounts then, or I'd just say quantities to get incorporated into the cell at the place of entry. So, so once you do that, yeah, just certainly follow through the same methodologies that you would as per the, you know, the protein oligonucleotide kit in terms of cleaning that up as per those instructions.
All right, thank you, Craig. Our time is up for the Q&A session. Thank you for attending the webinar. We will send you the entire recording tomorrow through email. And if we were not able to answer your question, we will personally answer your question at a later time. And have a great day. Thanks for attending the webinar.